All right. For the next presentation, I want to talk about the physiology of a muscle contraction. And basically, so yesterday in the previous video, I discussed the microscopic anatomy of muscle cells, and then now we're going to talk about all those components of muscle cells and how they actually work so muscles can generate movement. All right, skeletal muscles in specific. Now, before I get in this discussion again, there are a couple of good websites for you guys to take a look at. Again, I strongly encourage you guys, after you're done watching this video, to go to this website and check it out. Just because there are a lot of really good animations. So I'm going to explain to you a lot of mechanisms that occur at the microscopic level, but you're not going to see any of it in action. But on this website here, there's lots of very good visuals and actual animations for you guys to see. So I strongly encourage you guys to check out that site afterwards. And then again, the histology website, out of Delaware. This is again just an awesome web page to take a look at. Take a look at tissues. So on that note, let's talk about how muscles contract. All right. So basically what I want to talk about is I'm actually going to do this in a separate video, a review of membrane potential. Make sure you pay attention to that as well if you don't understand membrane potential. So, because that plays a big role in understanding this, plays a very big role in one of the properties that we'll discuss about muscle tissue in a second. And then, so in this video, I'll really start going into the major properties, and then talk about the nerve-muscle relationship, basically the importance of nerves and muscle contraction, and then basically what I mean by behavioral skeletal muscles, how they work, essentially how how the myofilaments that we discussed in the previous video interact with one another to actually create a muscle contraction. And then this is a very energy taxing process. Depending on time, I may not get into this in this video and do it in another one, but it's important to understand where muscles get their energy from, so they can continuously contract, whether you're trying to maintain posture or you're doing something rigorous like exercise. All right. So on that note, let's talk about the properties of muscle tissue, okay? Now, these properties I'm talking about here relate to all three types of muscle tissue, all right? Some more than others. Now, all three types of muscle are excitable, okay? Another way to think of excitability is irritability. They're irritable tissues, okay? So remember that every cell in the human body has a resting membrane potential. Okay, muscle cells are at about -90 millivolts. Okay, so basically what we mean then, when we say that these are excitable or irritable tissues, we're able to alter this membrane potential so we can generate a contraction. Now that's important because, as you know about membrane potential, we establish a membrane potential by creating a sort of imbalance across a cell membrane between certain ions like sodium and potassium. Okay, and that's why electrolytes are very important to our physiology, so these excitable, irritable tissues can perform their job. All right, that's why there's, electrolytes in Gatorade, so, when you're working out and you're sweating out all these salts, you can try to replenish them as quickly as possible. So, which is important to help you sustain exercise. All right. Now, when we say they're excitable or irritable, okay, muscle cells, muscle tissue can, most of the time we'll be talking about chemical irritation, but they could be sensitive to stretch and other factors that could cause electrical changes across the membrane, but primarily we're going to focus on chemical and neurotransmitters. So these are irritable tissues, okay? These cells alter their membrane potential so they can do their job, okay? So realistically, the irritable tissues of the body that you're going to learn about are muscle and nervous tissue, all right? And conductivity. Now, this is important because when I start talking about the nerve-muscle relationship, let's say here's a motor neuron innervating a muscle, and we'll say this is the muscle itself. Okay, now you'll notice that this motor neuron is only stimulating this little part of the muscle to contract. Okay, so when we remember, these muscle cells are very, very long cells. Like I said, they could range from being a couple centimeters in length to up to 20 centimeters in length and longer. All right, depending on, because remember, muscle cells run the entire length of the belly of the muscle. All right. Now, when we stimulate when a nerve stimulates a skeletal muscle to contract, you're going to see a local depolarization. So where the neurotransmitter is being released, okay, this area is going to be stimulated. All right. Now, what's going to happen then, though, is as this membrane potential changes here, the areas that are adjacent, the areas of cell membrane and cell that are adjacent are going to be sensitive to this. So then what's going to happen, just this change in electrical charge here will be enough to stimulate a change in electrical charge here. Then you can do the math. Then the more adjacent areas along the length of the muscle cell will be irritated. All right. So basically, instead of conductivity, we would say this action potential is propagated. Okay, or propagation is the term for that. Okay, so that's an important characteristic to possess, since a nerve doesn't stimulate the entire, doesn't innervate the entire muscle cell. It only innervates, small little parts, little sections of muscle cells. So we have to be able to conduct that wave of depolarization across the muscle cell itself. Okay, contractility. We've already talked about contractility, when we talked about muscle function. I mean, remember, when you hear the word contract, you should automatically think shorten. Okay, when you hear contract, shorten should be the first word that pops into your head. Because remember, when we took a look at sarcomeres the other day, remember, sarcomeres are the functional units of muscle. Okay, and they're composed of basically these thin and thick filaments. All right, so they're these alternating bands of thin and thick filaments. All right, remember, do you remember which are the thick and which are the thin filaments? Oops. Which are the thin, which are the thick? Remember, this would be actin, the thin filament, and then this would be myosin. Okay, and we said that, when you're going to see this more in depth today, that when these fibers interact with one another, because remember, there's heads on myosin that stick to actin, and then they pull actin towards the center of the sarcomere. Okay, so if you've got all of these myofilaments pulling towards the center of the sarcomere, you can already guess that the Z lines are going to shorten, and the sarcomere itself is going to shorten. And remember, when we looked at muscle cells, that they're loaded with sarcomeres. They're loaded with all these contractile units. Okay, so the entire cell is going to shorten. All right, so if we stimulate muscle cells to contract, the muscle itself is going to become shorter in length, and it's going to generate force and pull on a tendon so we can have movement take place. All right. And muscle cells are extensible and elastic as well. These are capable of being stretched, which is important because you have to remember, let's say here's a bone. Okay, so let's say here's your humerus, and then we'll say these are the bones of your forearm. All right, and we'll say this is your bicep muscle. All right, now remember that the tendinous attachments are going to cross the joints. All right, and remember, there's going to be a muscle on the opposite side as well. Okay, your tricep muscle. Now, you have to think about this, because when we want to create a certain movement, we obviously don't activate all the muscles in the area. If I want to flex my elbow, I'm obviously not gonna stimulate my biceps and triceps to contract at the same time. We would generate no movement if that took place. So what's going to happen, as this prime mover or agonist, actually that's technically not the prime mover. There's a muscle underneath here called the brachialis. So that would actually be the agonist. The bicep is what we would call the synergist. It assists, okay? So an agonist is a prime mover. When I say that, I mean it's the muscle that's doing most of the work, and then a synergist is assisting the prime mover. So basically, these muscles are working together. So basically, they're contracting, and their tendons are pulling on the bones, that are across from the joints. And then what's going to happen? That's going to make your forearm curl upwards. You're going to flex your forearm. Now, what do you think is going to happen to this bicep muscle? obviously, if these muscles are shortening, this muscle, this tricep muscle, is going to lengthen. Okay, that muscle is going to lengthen and it's going to be stretched. All right, it's going to be stretched. And then if we want to, then if we want to get the elbow back to its, back to a straight arm position, then the tricep muscle would contract and then it would shorten, and then these muscles would then, lengthen as well. So muscles are always shortening and lengthening depending on what types of movements we're trying to create. Okay, so keep that in mind. And being that the tricep muscle works against the bicep and the brachialis, this is what we would call an antagonist. Okay, that's what we would call an antagonist muscle. All right, so keep that in mind. And then elasticity, you have to remember the concept of elasticity, that whenever we apply pressure to an object and stretch it, okay, once the pressure is relieved, it's going to recoil back to its original shape. Okay, so keep that in mind. So these are all the various properties of muscle tissue that allow it to do its job. It's irritable, it's able to conduct, it's able to propagate this wave of depolarization across the cell. Muscles contract and shorten, that's where the movement actually comes in. And then, like I said, because not every muscle is going to be working here, the muscles that will be resting are going to be stretched, hence the extensibility. And, then elasticity, again, if you apply pressure to an object and stretch it, when the pressure is relieved, it returns back to its original shape. Okay? So now when we want to talk about a muscle contraction, I like to break this down into 4 separate phases. Okay, excitation, contraction, coupling, contraction. Okay, so these first 3 stages. Okay, these first 3 stages are obviously involved in the contracting muscle. Okay, so these need to take place in sequential order for the muscle to shorten, for it to actually contract. Relaxation obviously occurs after these have taken place. Okay, but relaxation is important, because muscles have to recoup, they have to recover so they can contract again. All right, so I mean, technically this isn't really a phase of contraction. This is recovering from it, but I'm just throwing this in there. All right, so now let's break down these phases of a muscle contraction individually, step by step, throughout this entire process. All right, so now in order to understand excitation, we have to understand how the muscle gets irritated. All right, now when it comes to skeletal muscles, okay, skeletal muscles are innervated by motor neurons. Okay, so remember the term innervate. Okay, when I say innervate, I'm talking about a nerve plus a tissue. Okay, now you have to remember, nerves innervate more than just your muscles. I mean, there are nerves innervating pili erector muscles in your skin. There are nerves that are innervating glands. Okay, there are nerves. So basically, whenever a nerve branches out and communicates with the tissue outside of the central nervous system, we would say that that nerve is innervating that tissue. Okay, and one thing you have to bear in mind is that yes, we have voluntary control over skeletal muscle, but without the muscle being innervated, it cannot contract. Okay, you need that nerve to make it contract. Okay, it's not like cardiac muscle or smooth muscle that have a lot more unstable membrane potentials and that have their own rates and rhythms of contraction. Skeletal muscle is dependent on nerves. All right, I mean, I don't want to say a good example, but a relatable example to this. Think about someone who's paralyzed, someone who's been in a traumatic accident and has had their spinal cord transected. Okay, so let's say this individual had their spinal cord transected. Let's say this was at about, I don't know, L3, lumbar vertebrae 3. Okay, now obviously I mean, you're going to think of their legs in this situation. All right. Because you essentially cut these wires or cut these nerves, okay, you're not going to be able to get nervous impulses or action potentials down past this area where the wires have been cut. Okay, so as a result, you lose function of the muscles of the legs. You cannot use those anymore. All right, and then what happens to the legs? They begin to atrophy. Okay, that's what happens when you disuse muscles. The proteins break down. Okay, and then you use those amino acids and proteins for other purposes. There's no sense in maintaining the development of a tissue if you're not actually using it. Okay. So remember, nerves innervate muscles. Okay. And, again, the nerves that innervate muscles for the purpose of a contraction are called motor neurons. Okay, they're called motor neurons. All right, and as it states here, they cause a local disturbance of the membrane potential. Okay, so they irritate the muscle tissue. And they do this by releasing a neurotransmitter called acetylcholine. Okay, acetylcholine. Now remember, when nerves communicate with other tissues, you have to remember that nerves have a cell body, and then they've got these long branching axons, and then at the very ends of the axons, there are these boutons or bulbs. Bouton is a French word for button. That's what they some people call these. All right, these are what we call synaptic terminals. Pardon the handwriting, folks. These are what are called synaptic terminals, okay? And then within those synaptic terminals, there are specific neurotransmitters that are released, okay? So when a nerve generates an electrical impulse down its axon, the whole purpose of that is to stimulate these synaptic bulbs or these synaptic terminals to dump out their neurotransmitter, and then the neurotransmitter then binds to its receptors on whatever the target tissue is, whether it's another neuron, a gland, or a muscle or muscle cell. That neurotransmitter is going to elicit some kind of response by big part by causing a disturbance of the membrane potential, and you'll see that more in depth in a second. Okay. Now, when we want to talk about how muscles are innervated, we have to take a look at what are called motor units. Okay, what a motor unit is, it's a somatic motor neuron. Okay, with all the muscle fibers it innervates. All right, so let's kind of jump. So let's kind of jump and take a look at this picture here. So a somatic motor neuron. Okay, so remember, motor neurons. When you hear motor, okay, you should be thinking muscles or glands. Okay, so motor neurons are the neurons that branch out from the central nervous system and stimulate our tissues to perform some kind of an action. Okay, and as you can see here on this this transverse section of the spinal cord, the motor neurons exit anteriorly. It's something you got to bear in mind, that motor neurons. So basically, output is always leaving the spinal cord from the ventral or anterior side, and then we'll go with ventral. And then sensory input is always coming in dorsally, okay? So that's something to bear in mind. So sensory dorsal, okay, motor output, okay, ventral. All right, now, so basically the bodies of the neurons are essentially going to be found within the spinal cord, and then their axons are very long. They're going to project out of the spinal cord towards the muscle. All right, and then these nerves, what they're going to do, so you can see this broken down here. Remember the parts of a muscle. This would be the belly. Okay, this would be a fascicle, and then these would obviously be the myocytes. Okay, the muscle cells, as we discussed in the last video. All right. Now, so you can see here that a motor unit essentially is, so you can see the axon of this neuron is gonna be embedded within the connective tissue spaces of the muscle, and then it's going to start to branch out to different muscle fibers, okay, or muscle cells. All right, so a motor unit is basically one axon or one nerve and all of the individual muscle cells that one nerve innervates. Okay, that one nerve innervates. That's what a motor unit is. All right. How many muscle fibers or muscle cells are innervated on average by motor units? That varies greatly depending on the muscle. All right. It could vary from 80 to 100 muscle fibers per motor unit. All right. It could be 3 to 15. A lot of it depends on the size of the muscle fiber and the force of contraction that we need to occur. Okay, so bear in mind that's what a motor unit is. Okay, so think about this for a second. So let's say we're looking at an ocular muscle versus, I don't know, a quadricep muscle. Which one would have more motor units? Which one of those two do you think would have more motor units? Which one do you think? The ocular muscle or the, when I say ocular muscle, I mean one of the small muscles of the eyeball. One of the small muscles of the eyeball. So think about this. Which is the bigger muscle? Which is the bigger muscle? Which one is going to be generating more force? Okay, so if you're guessing the quadricep muscle, all right, you are correct. All right, the quadricep muscle is obviously going to have more motor units. You're going to have to recruit, in order for a quadricep muscle to contract, if you want a quad to do the kind of work that it normally does, you're going to have to recruit more muscle fibers at once. If I want to try to extend, 130 pounds, with, while in a sitting position, you're going to have to recruit more muscle fibers within that process. So there will be more motor units for more power generation. Okay, you don't need a ton of motor units. Within these small ocular muscles, you don't need to move your eyeballs at the same force that you would need to contract a quadricep muscle. Okay, because one thing you have to bear in mind is that about 1 square centimeter of muscle cell, okay, about one square centimeter of muscle can generate quite a large amount of force. Okay, quite an amount of force. So let's say that would be about, we'll say 3 to 4 kilograms of force per centimeter squared. Okay, so a quadricep muscle has the potential to generate about 800 pounds of force. Okay, if we stimulated all the motor units within a quadricep muscle, that those muscles could generate 800 pounds worth of force. Okay, now that's not going to happen because that would rip our tendons clean out of our bones. Okay, but the beauty of being set up like this is that when we do need to increase the amount of force, we can recruit more motor units into the process. Okay, so that's something to kind of think about and keep in mind. So that's a motor unit. Okay, so again, a neuron and all of the muscle cells that it innervates. All right. And like I said, you can see why then if someone is paralyzed, if they have the spinal cord damaged, they're unable to get action potentials or impulses down, okay, down and out. All right, because you have to remember, when you want to create a movement to contract a skeletal muscle, we can technically say the contraction begins in the brain because you have to plan this process out, okay? And then you send that information down the spinal cord, and then it exits out the ventral aspect of the spinal cord out to the muscles, okay? If you're unable to get that message from your brain out to the muscles, you will not be able to move. Okay, and then here are just some other images of motor units. Here's more of a close-up, so you can see right here this nerve coming in, and then all the muscle cells, and it's innervating. It's kind of cool, actually. So then we can take a look at this here off of the histology website. So again, you see this nerve coming down, and then you see the synaptic terminals branching off from the main axon. And then, now these right here, this is what we would call a motor end plate. Okay, motor end plate. Okay, now we're going to talk about this in a second, but this motor end plate is going to form what's called the, I'm just going to abbreviate this for now, is what's called the NMJ, or the neuromuscular junction. Okay, because there are certain events that take place here. So once that electrical impulse, that action potential from the nerve, generates its way down to this motor end plate, then neurotransmitter is going to be released, and then will depolarize that area of muscle. All right, so basically, so you can see these various motor end plates where they're going to be stimulating the muscle to contract. So keep that in mind. And then right here, here's an electron, here's an electron microscope image of this. This is just awesome. So you can see the muscle fiber here. You can see the motor end plates. All right, this is just great. You can see the branch. So here's the main axon. Okay, and then you can see here the synaptic terminals branching out to the muscle cells that we need to stimulate to contract. This is really cool. All right. So now let's talk a little more in depth about the neuromuscular junction and how this excites the muscle tissue. All right. So again, you've got the axon. You got the electrical impulse, the action potential traveling down the axon, and then it's going to start to branch out. to the motor units. Okay, so then what I'd like to do now is take a look at this neuromuscular junction a little more up closely. All right, so again, you can see the muscle cell here. Let's review this again for a second. You can see, multinucleated. So, okay, you can see it's multinucleated. You can see all the myofibrils, the muscle fibers within the cell. Now you can see the sarcolemma, which is the plasma membrane, and then you can see the sarcomeres, the alternating light and dark patterns here. Okay, these are the actual contractile units of the muscle. All right, so now what's going to happen? Come on. What's going to happen then is there's a series of events that takes place at this neuromuscular junction. So again, that electrical impulse, that action potential is going to travel down to this bouton or this bulb. Okay, and what that's going to do then, that electrical disturbance of this synaptic bulb is going to stimulate the opening of calcium gates. Okay, and then calcium will diffuse into the into this the very end of this nerve cell here, and what that's going to do is that's going to stimulate these little structures in here called synaptic vesicles. Okay, now if we take a look at kind of the microanatomy of this synaptic vesicle, what you're going to see here is you're going to see this bulbous structure, this button-like structure. That's why they call it bouton again. That's what they thought it looked like when it was first described under a microscope. Okay, and then inside of this bouton or this bulb, you're going to see these little sacs, or these synaptic vesicles is what they're called. Okay, and then within these sacs, that's where neurotransmitter is stored. Okay, and depending on the type of motor neuron, it's going to store a different neurotransmitter. Okay, and again, remember the neurotransmitter in this situation is going to be acetylcholine. All right, so then what happens, that calcium diffuses inward, and then there will be a series of events that take place. But what will happen then is that'll stimulate these synaptic vesicles to fuse with the membrane of the synaptic bulb. Okay, and then you can see what's going to happen then is once these vesicles fuse with the plasma membrane, then they'll open up, and the neurotransmitter is released. Remember, this is what we call exocytosis. Okay, exocytosis. All right, now, and then so now what you're going to see, you'll notice that this is a common misconception people have early on as students is they think that these nerve fibers actually physically touch the tissues they innervate. They don't. Okay, there's a small little space. Okay, there's a small little space in between where the nerve meets its target tissue that's called the synaptic cleft. The synaptic cleft. Okay, so you got this synaptic cleft here. Now what's going to happen is as these vesicles start to unload, okay, as these vesicles start to unload their neurotransmitter, you can do the math on what's going to happen. What direction is the neurotransmitter going to travel? It's going to travel down its concentration gradient. It's going to diffuse down towards the skeletal muscle cell itself. And this picture doesn't really do this process a lot of justice. Okay, this does not really do this a lot of justice at all. All right, just, I say that because there are so many more synaptic vesicles here that, that you can see, and there are so many, and there's so much more acetylcholine involved with this as well. Okay, so normally when you would be watching this process in action, well, you would see these vesicles fusing, and then you would see thousands and thousands and thousands of acetylcholine neurotransmitters diffusing down this synaptic cleft towards the membrane of the muscle cell. Okay, and then what's going to happen is acetylcholine is then going to bind with its specific receptors. Okay, it's going to bind with specific receptors, and essentially these receptors are gates. They're sodium gates. Okay, so they're ligand sensitive gates. Okay, now when we say ligand, what we mean when a chemical message binds with this receptor. Okay, now you'll notice that there are these kind of inward folds, or these invaginations of the sarcolemma, where the neuromuscular junction is, where these motor end plates meet the muscle tissue. Okay, now these inward folds are called synaptic troughs. Okay, and what these essentially do is they increase the surface area. Okay, and I'm just going to abbreviate surface area. Okay, they increase the surface area of the muscle tissue. So the beauty of this is if we have more surface area, there's more room for receptors. Okay, so the more the receptors we have here, all right, the easier it's going to be to excite or irritate or stimulate this muscle tissue to contract. Okay, so that's something to keep in mind. So that's essentially the first part of excitation. The action potential travels down the motor neuron. We stimulate the calcium channels open, calcium diffuses in, and then that stimulates these synaptic vesicles to fuse with the with the membrane of this motor neuron, and then of the synaptic bulb, and then via exocytosis, acetylcholine will be released and then diffuse down its concentration gradient across the synaptic cleft and bind to its specific receptors. All right. And as long as acetylcholine is bound to its receptor, this muscle tissue will continue to be irritated. Okay, so obviously, remember when we talked about membrane proteins, we also talked about enzymes. Okay, because we don't want to overstimulate a tissue because that could damage the tissue. All right, and the enzyme here is going to be acetylcholinesterase. Acetylcholinesterase. Okay, so essentially, what's going to happen then? I'll talk about relaxation later on. But when we want this muscle to relax, acetylcholinesterase will separate acetylcholine from its receptor, and then it'll essentially cleave it into two molecules, essentially acetic acid and choline, and then a process called reuptake will take place, and this motor neuron will suck up the leftover acetylcholine and remnants, and it will put it back together and then repackage it in these synaptic vesicles to be used again. Okay, so that's part of this process of the neuromuscular junction as well, the reuptake, getting the acetylcholine out of there. All right, so now once we cause this local disturbance, in the membrane potential of this area, once we depolarize this area, what's going to happen then is that signal has to propagate along the length of the muscle cell itself, as you can see the beginning of that here. All right, so what's going to happen then, remember that acetylcholine will be released, and remember these receptors are essentially sodium channels, okay? Because remember, there's more extracellular sodium than intracellular sodium here. So then what will happen is as sodium diffuses inward, okay, so if we were graphing this, okay, and let's say this were at negative 90 millivolts, all right, and we were releasing acetylcholine, and let's say this would be 0, this would be positive 60, all right. Now what's going to happen is as we increase the amount of acetylcholine, you're going to see the membrane potential become more positive. All right, because remember, this charge is negative 90 millivolts. As you have more positively charged sodium diffusing inward, okay, it's going to become more positive, and then you'll see a big boink, a big up spike, and then because that'll cause a greater influx of sodium, and I'm not going to go that in depth with it right now. All right. But what will happen then is as we have this influx of sodium in this local area, that'll be enough to cause a disturbance in the adjacent cell membrane. Okay, so then these voltage sensitive gates. Okay, so basically as the membrane potential or the electrical charge changes, okay, these voltage sensitive gates that'll create a conformational change. Remember, these gates are essentially just coiled proteins that are embedded within the cell membrane. Okay, so what'll happen is they'll cause some uncoiling and untwisting of these proteins, and then more sodium will diffuse in. And then you can do the math. Then this adjacent cell membrane will become depolarized. And then the next adjacent, and then same thing in that direction as well. Okay, so that's essentially excitation, irritating the muscle cell, okay, at the neuromuscular junction. All right, and then that's going to lead into the next part, the beginning of excitation contraction coupling, which I'd like to call, we'll just abbreviate it, but abbreviated ECC. Okay, so now this is where that triad comes in. Remember, the triad is the T tubule plus the sarcoplasmic reticulum, okay? Because you can see here, you see sarcoplasmic reticulum on both sides, and then the transverse tubule right there. And remember, this transverse tubule is an invagination or long inward folding of this plasma membrane or sarcolemma down into the muscle cell itself. All right, and then you can see the sarcolemma, or I'm sorry, it's the sarcolemma, the sarcoplasmic reticulum. Remember, it's a specialized form of smooth ER, and remember that the sarcoplasmic reticulum is where we store calcium. That calcium is essential for a muscle contraction. All right. And then you can see how these structures just overlap the sarcomeres themselves. Okay, so you can see the contractile units underneath here. You can see the myosin. You can see the actin. This would be that bare zone or the H zone within the center of the sarcomere. Okay. So what's going to happen then, as this action potential is propagated along, that action potential will travel down the transverse tubules. Remember, we talked about this influx of sodium, right? Okay, so then there are gates that are located all along, that are basically in between these transverse tubules and the sarcoplasmic reticulum. All right, so as that wave of depolarization propagates along the sarcolemma or the cell membrane, it's also going to travel its way down the transverse tubules, and then that wave of depolarization will basically cause these calcium gates to open. Okay, these calcium gates are now going to open. Okay, and then calcium will be released from the SR, and that calcium will then diffuse down into the sarcomeres. Okay, the calcium will diffuse down into the sarcomeres. All right. Now let's review why this is important again, because you have to go back and remember. You have to remember the differences between actin and myosin. Okay, so let's say you've got. So let's say this is, shoot, I'm drawing this poorly. Now remember, actin has these binding sites. Okay, they're specifically called active binding sites. Okay, and remember these active binding sites, remember, this is where myosin interacts with actin. Okay, because remember, myosin, that big thick protein, all these coiled myosin filaments are wound together, and then there are all these heads that come off of the myosin filament. Okay, and then remember that there is actin, kind of forming in a hexagonal three-dimensional fashion, okay, overlaps the myosin filaments, okay? But remember, resting actin has regulatory proteins that are covering these binding sites. Okay, remember troponin and tropomyosin. Okay, so remember that, so let's say we got all this actin here, and then we've got this tropomyosin kind of coiled around the active binding sites, and then we'll throw a troponin in there. Okay, so essentially what's going to happen then is, as calcium diffuses down into the sarcomere, that calcium is going to bind with troponin. Okay, and then what's going to happen, that's going to cause troponin to take on a conformational change. It's going to twist and coil in a different shape, and then what that'll do is that'll make tropomyosin unwind and uncoil off of those active binding sites. So now those binding sites will be exposed. Okay, so as long as there's enough calcium available, okay, and within the muscle, and these and it's bound to troponin, these active binding sites are exposed, we can contract muscle. Okay, if we start to run low on intracellular calcium, then that muscle is going to be unable to contract. Okay, so you have to keep that in mind. So that's essentially what excitation-contraction coupling is basically setting up the myofilaments for the contractile processes that are going to take place. All right, so they essentially get these regulatory proteins out of the way so actin and myosin can now interact with one another. All right, which leads us into the next part, the actual contraction. Okay, the actual contraction. Now, when the contraction takes place, remember yesterday I talked a little bit about the myosin head. Okay, remember I said on the myosin head there's an enzyme called ATPase. Okay, there's an enzyme called ATPase. All right, and then there's also a site on the very end of the myosin head that will interact with the active binding site of actin. All right. Now remember, we talked about yesterday when I introduced muscles and their the property of contraction. Okay. Remember when we contract a muscle, we're taking chemical energy, ATP, and converting that into mechanical. Okay, and in order to do this, we have to cleave some of the chemical bonds within ATP so energy can be released. Okay, that's where ATPase comes in. So essentially, so what's going to happen then, this would be the bound calcium on the troponin, okay, and then you can see this tropomyosin, I'm just going to abbreviate this, tropomyosin has now been unwound and these active binding sites are now exposed. Okay, so now we're ready to go. So then what happens is once this, so now this myosin head, obviously you can see that it is not interacting with actin yet. Okay, so what happens is this would be an ATP, and then this would actually technically. So what's going to happen is when we want to get this actin to get up there and bind with and interact with actin, what we have to do, we have to hydrolyze ATP. Okay, so what this essentially is, this is ADP and a phosphate. Okay, ADP stands for adenosine diphosphate. So what we did is we cleaved off one of the three phosphates. Okay, and what that'll do is that'll make this head kind of cock in a certain fashion, and then we'll have then there'll be energy for it to actually move upward and then bind to the bind to the active binding site and interact with actin, which is what you can see right here. Okay, so now these two are bound together. Now these two are bound together, okay? And this is what we call, this right here is what we call a cross bridge. Okay, this is what we call a cross bridge. When actin and myosin are actually interacting with one another, we form a cross bridge, okay? Now, what's going to happen then, so now these are actually bound together and they're interacting with one another, but there's really no movement taking place yet. Okay, there's really no movement taking place yet. So you can see right here zoomed in. So what's going to happen next then is you can see what's happening here is then after this hydrolyzed ATP, actually so now ADP and then this phosphate are released, and then the energy from that hydrolysis and release will then make this head undergo a conformational change. Essentially, this head is going to cock a little bit. Okay, think of it like a hammer of a gun cocking. Okay, and then as this myosin head kind of cocks a little bit, it's going to pull on the actin. Okay. Now you have to bear in mind that you see all these various these myosin heads here. Okay, there are going to be many myosin heads at once interacting with these actin filaments. Okay, interacting with these actin filaments, and then remember that, and then these heads are cocking in the direction towards the center of the sarcomere. Okay, and so you remember what happens when a muscle contracts. The sarcomere gets shorter. The Z lines are pushed closer towards the center. All right, and this is what we would call the power stroke. Okay, that's what we would call the power stroke. All right. Now the next part of this, so now this myosin head has cocked and it's pulled on the actin. Now we have to get this. We have to essentially reset this so this myosin head can do this all over again. Okay. Now, in order for this to happen, what a new ATP. Okay, new ATP has to bind to this head, and then that separates this cross bridge. All right, and then the process is going to start all over again. This ATPase will then hydrolyze. Will hydrolyze this ATP molecule into a new ADP and PI. phosphate. All right, and then that will get this head in position, allow it to reconnect with the actin, and then the ADP and phosphate will be released. That'll make the head cock, the power stroke again, and then a new ATP molecule will come and bind, and then bang, the myosin head will return back to its resting state, and you'll keep cycling through ATP. You'll keep cycling through, ATP and phosphate. Okay, you have to remember, within muscle, there's a lot of ADP stored within muscle. Okay, so basically, we're constantly recycling ADP within muscle, and we're constantly generating it as well through different forms of metabolism, which I'll save for later on. All right. Now, as long as there is ATP present and calcium present, enough ATP, I should say, and enough calcium present, we can continue to undergo this process of what we call cross-bridge cycling. Okay, cross-bridge cycling. Okay, now you have to bear in mind, though, when these muscles contract, okay, as this actin is pulled towards the center, and all these sarcomeres within the filaments contract, the muscle the muscle twitches. Okay, so this would generate a muscle twitch. So you would see this muscle cell kind of go doink towards the center, and then it would stretch back out again, and then it would go doink back to the center, and then go back again, and it would go doink, and then it would keep twitching all the time. All right, it would keep twitching all the time. So a little bit of clinical application to all this process. Now, let's say someone is, you ever heard of the disease? And this kind of sums all this up in a nutshell here. So everything we just discussed. But let's do a little clinical application. I'm going to abbreviate this. Myasthenia gravis. Okay, myasthenia gravis is an autoimmune disorder. Sorry, talking and writing isn't always easy sometimes. It's an autoimmune disorder. So what they found with this is an autoimmune disorder that takes place at the neuromuscular junction. Okay, so let's say we got muscle here. Now what they found is that people develop what are called autoantibodies. Remember, auto meaning self. Okay, so if you develop antibodies against yourself, that means your immune system is targeting one of your own tissues. That's not good. Okay, and what these autoantibodies do is they bind to acetylcholine receptors. Okay, they bind to acetylcholine receptors. And what they do then is they block acetylcholine. They block acetylcholine from binding to its receptor. All right, so now what's going to happen is you're going to be unable to excite this muscle. You're going to be unable to release calcium into the sarcomere and expose those active binding sites. All right, and so you're not going to be able to allow actin and myosin to interact with one another. Okay. So what's going to happen then, a person clinically is going to start to present with some muscle. It'll typically start out as general muscle weakness. Okay, you might get fatigued walking up a set of stairs or just doing normal daily functions. And then what you'll start to see are, you'll start to see some droopy facial muscles, especially muscles of the eye, those ocular muscles. Typically when people have muscle disorders like this, the small muscles are always affected first. Okay, and then what'll happen is they'll test you for this. They'll test for the autoantibodies, but then when they treat you for this, one of the medications they'll give you is an acetylcholinesterase inhibitor. Acetylcholinesterase inhibitor. Now think about this. Why would you want to do that? Remember the function of acetylcholinesterase. These enzymes that are found near receptors. Okay, remember these enzymes, they unbind chemical messages from receptors. Okay, so then as a result, okay, what you're doing then, and remember what these do, what this enzyme does is it cleaves. Remember, it breaks this molecule, this acetylcholine, into two separate molecules. Remember, acetic acid and choline, and then that's basically useless. So if you inhibit this enzyme, you increase your acetylcholine levels, and you increase the chances of acetylcholine coming in contact with this receptor. Okay, and then you have to go from there with the treatment. All right. You know, people have calcium deficiencies or severe calcium issues. You're going to see, you know, think about what would, I'm not going to explain this. I want you guys to think about this. What would happen if someone had calcium problems, if someone was hypercalcemic or hypocalcemic? What would happen if people's calcium homeostasis was thrown off? All right. You know, okay, so I mean, one of the, I mean, few but many clinical applications that can go towards this, and I did forget to talk about the relaxation aspect of this. I apologize about that, but relaxation is relatively simple. Basically, you take everything I just talked about and go backwards. Okay, so essentially, the major phase of relaxation is the return of calcium into the SR. Okay, so there are going to be these active pumps that will pump calcium out of the sarcomere back to the SR, and then you can do the math. The tropomyosin will then cover these active binding sites, and then now myosin and actin cannot interact, and now will become a relaxed muscle. And then on the excitation phase, ACH will be broken down, and then will be taken back up by the neuron as well. So essentially, relaxation is easy. You just go backwards. Okay. So again, here's the physiology of a muscle contraction, kind of a very brief, basic explanation of this. Later on, like I said, I'm going to do a separate video on membrane potential just to review that. And then later on, if time permits, I'd like to make a video talking about factors that go into the force of muscle contraction and muscle metabolism as well. If I don't get to do that soon, I will do it later. And again, if you folks have any questions, don't hesitate to ask. And go Packers in the Super Bowl this weekend. I am from Wisconsin, so I've got to support my boys. Good luck.